Well, welcome everybody to the Boston Children's Hospital Gene Therapy 10th Anniversary Celebration. I'm Colleen Dansereau, and I'm the Director of the Clinical Operations for the Gene Therapy Program. And I've been fortunate enough to be a part of this since its very beginning, actually. And I've had the privilege to observe many milestones and life-changing moments in our patients' lives. I hope you enjoy the program we've planned for you today. If you have any questions, please type them in the Q&A box in the bottom, and we'll answer those during the QA session at the end of our program. And now I'd like to turn it over to Dr. Dan Bauer, the new director of our program, who was recently named the Dagris Family Chair in Gene Therapy. And we'd like to give a special thanks to Trust Board member Todd Dagris and his wife Elizabeth for their very generous gift. We in the Gene Therapy Program are deeply grateful for your support of our work. Dan?
Thank you, Colleen. Likewise, I want to thank the Dagris family for their fantastic support. I'm really excited to take the reins and build upon the existing gene therapy program, which has treated more than 70 patients from 20 countries through 19 gene therapy clinical trials. I'm a hematologist and molecular biologist who became interested in gene therapy when I realized the time has now come when we can realistically apply new tools like CRISPR technologies to address devastating diseases at their root cause. What I'm especially enthusiastic about is how our gene therapy program focuses on what we call translational research, which is how can we marry the fantastic basic science and clinical care that we have at Boston Children's Hospital to move the science from the laboratory to the bedside and then bring lessons from the bedside back to the lab bench, while taking meticulous care of each of the important steps in between. So this is an opportune time for the field as it's broadening to include new possibilities like gene silencing, gene editing, base editing, and more. What ultimately inspires me is the stories of patients. So I'm going to conclude so we have plenty of time to hear and reflect upon their stories. All of this is only possible because of our institution's vision and commitment to innovation. So with that, it's my great honor to introduce the CEO of Boston Children's Hospital, Sandi Fenwick.
Thank you so much, Dan. It's really, truly an honor to be part of this celebration, the 10th year anniversary of this really, truly pioneering program. I want to thank so many people for joining today. And most importantly, to so many of you who are here who will have a little bit of a speaking part. You know, I have spoken a lot lately, even more so than usual, and asked what is the best part of my job? And there's such an easy answer. And Dan, you used it. It is the inspiration that is part of this hospital. And the inspiration comes from all of you. You clinicians, you physician scientists, nurses, nurse researchers, and of course, our patients and families. You are absolutely never satisfied with what you know, what you can diagnose, and what you can actually then do something about and treat. You make the impossible possible through discovery science, and then translation, and then clinical trials, and then treating patients sometimes for the first time. The discovery science, the tenacity, the perseverance, the devotion, the resilience that really is embodied in every one of you. And then you marry that with real compassion, real care and compassion for patients and families. And of course, there's the patients and families. Your love and hope for your child, the trust that you put in our team, and the hope that one day there's going to be an answer and a treatment, and hopefully one day maybe even a cure. This is what makes Boston Children's Hospital the extraordinary place that it is. The gene therapy program is really a shining example, built on the work and the science of over 2 decades, and based on pioneers like Stu Orkin, trailblazers like David Williams, who came back to Boston Children's Hospital and built this program with Colleen Dansereau and Sun-Yun Pai. Current leaders, Dan Bauer and Christy Duncan. And of course, the whole team that I'm sorry I won't mention every one of you by name, but you've brought new hope and relentless pursuit to treatments and hopefully, as I said, one day cures for so many patients who are still waiting. So I can't thank you enough for what you have done, and I know you will not stop. And you really, truly will not stop until every child is well, as we say. So congratulations on this really, truly wonderful anniversary. And I know this is only the beginning. Thank you.
Thank you, Sandy. And now I'd like to introduce the rest of our gene therapy team. And as I mentioned, I'm Colleen Dansereau, and I'm the Director of Clinical Operations for the program. My role and responsibility is I'm really, truly behind the scenes, making sure that you all have, our team has what they need to conduct the clinical research, both from the, Dan said, from the translational sense, from the lab to the patient. I like to think of myself as the woman behind the scene that moves the levers, kind of like the Wizard of Oz. But I'd like to introduce the rest of the team. So David Williams, Dr. Dan Bauer, Dr. Christy Duncan, Pietro Genovese, Christian Brendel, Brenda McKinnon, Amy Federico, Pauline Feinberg, Emily Morris, and Bridget Kerwin. And I'd be remiss if I didn't also give a shout out of thanks to the ever-growing village within Boston Children's that is instrumental in allowing us to do our work. Notably, the translational lab, the therapeutic apheresis unit, our inpatient and outpatient clinical care units, our pharmacy, our cell manufacturing partners at the Dana-Farber are all key partners with allowing us to do our work. We have 3 interviews for you today, 2 of which were pre-recorded. The first highlights our gene therapy trial for adrenoleukodystrophy. This was the first pharma-sponsored trial in gene therapy at Boston Children's. Dr. Christy Duncan, the trial's principal investigator and our newly named medical director for clinical research, will interview the Elliott family from Ireland. Their 11-year-old son, Matthew, was the first in the world to receive gene therapy for adrenoleukodystrophy. Adele and Damien Elliott chose gene therapy over a stem cell transplant after their first son, Mark, who also had ALD, had undergone a stem cell transplant and unfortunately died of the disease.
If you would, like, how, like, what did it take, like, just to get you on a plane to leave your life for another, like, for months to come here? Right? I know, and your legs probably fell asleep on that plane because Lee was never a small boy. And like, I know, like, what was it like? Like, what was that decision like, and how did you do that? And what were you feeling?
To be honest, it was I think more or less what we've been through with regards to Mark. Whereas with Mark, it was because we got so much told to us that it was so minute of a chance that it would take, and we hemmed and hawed whether it was the right path. Whereas this, it was kind of like felt right, and we didn't even, I don't even think we had a long conversation about it. We just kind of, yeah, this is what we're doing it. And the minute that we found out that something, you know, was in the pipeline, it was just like, right, we're getting on to this, we're finding out what we have to do. And then when it was that he was an ideal candidate, it was just like,
it was meant to be.
It was a no-brainer, you know. It was something that, yeah, didn't even have to question it. We're just doing it. And if you had said no, we would have fought with you.
Yeah, I never would have.
We would have not have left Boston until we got the right answer. That's just the way it was.
And so I think you knew at the time, but you knew that Matthew was first, right?
Yes, we knew he was going to be first.
And that didn't make you nervous at all?
Not really. We weren't really nervous. Now, the only thing we were nervous about was, do you know the 2 weeks where you first, well, it was 4 weeks, wasn't it? We came home for 2 weeks.
Screening.
The first 4 weeks when we were over and he had to go through, you know, all the testing.
Yeah.
That was the nervous time because we wanted him to be accepted. So it was nervous that something would come up that he wouldn't be accepted, not nervous about the gene therapy. We couldn't wait to get it started. We just had 100% faith in it that it was going to work.
You know, that's really interesting, and that's good for me to hear, because I think at the time, from our perspective, most of those tests that we do, we knew we're going to be fine and that you were going to be able to get in, because the only thing that I think could have held back was the MRI. We had the MRI so recently. So I think that's probably just good learning for me too, just to explain to people that those screening tests are so unlikely to get you off that study. A lot of families are really anxious, understandably, with the chemo and coming into the hospital and getting chemotherapy for your son who's never gotten, has no reason beyond this to get chemotherapy. How did you feel with that? How was that whole process?
I think again it was the fact of going through Mark's chemo, and Mark had such aggressive chemo because he had the adult chemo. I knew it came across that he wasn't going to get most of the stuff that Mark got, and it was less aggressive and there was less.
No GVHD.
It was kind of more of a relief. And then, of course, then going through the chemo, it was just like, when
just defied you.
Yeah, when is it going to come on? Because of being with Mark, it was like, right, when's the sickness? When's the hurt? When's the side effects coming? And the kind of.
He didn't really take many.
No, he was very active, if I recall. I think he kept our hallways very busy.
I came to visit him.
I remember, I think I'm remembering soccer/football being played in the hallway quite a bit during transplant, if I'm remembering accurately. Yeah. And how about, is that whole time a bit of a blur, or the day the cells went in?
Some of it, yes, but that was for different reasons, because obviously when his cells went in, my mummy had died at the same time. So that was kind of hard along that lines, but it kind of overtook that, although obviously we were still upset and still sad about that. It was like because we still celebrate it today. Matthew has a rebirthday every year. So that was his rebirthday, and I was like, this is the start of his life. He can have a nice normal life. So we kind of took it in like that, but I was still expecting him to be a lot more sick than what he was. Like, he was so good with it, and he just took it and got on with it.
Matthew, what do you think about all of this?
I'm lost for words.
That's okay. You don't have to be.
He's not usually lost for words.
No, I know that. I remember. I remember the personality. I remember all of those pieces. And so I know the answer to this question, but I'm going to ask it anyway. Would you do this again? Yeah.
In our next interview, Dr. Erica Esrick, the principal investigator for our sickle cell gene therapy clinical trial, will speak with Lydia Masula and her 14-year-old son, Matthew. Matthew received gene therapy for sickle cell disease in October of 2019.
For both of you, I guess, what was it about your sickle cell that made you want to look for a treatment like this?
Well, I didn't really think about, I knew I had sickle cell, but I didn't think much about it. I just, did stuff like anyone else would do. But I knew I had it, but I didn't think too seriously about it.
Yeah. I think for us as parents, it's just what you do as a parent. Having seen Matthew fight through it to a point, to a certain point where at one time he was in the ICU, so you kind of look at the bigger picture, when he's older, what if the port doesn't work, then what next? And so I think that's why we thought about different options rather than just relying on the exchanges.
Yeah. So Matthew, how has life changed for you since gene therapy?
Well, I went out for school for a couple of months, then we went back inside. But So it's like when I was in school and like when I did PE and stuff, I felt like I had more energy. But since I kind of came back inside, I exercise once in a while, but I'm mostly sitting down, so I can't really tell you. Like, I can't fully answer that question yet. I'm active, but like mostly sit down.
Well, it's a funny question too because of COVID right now. So many people's lives aren't exactly normal right now. I know that.
Yeah. Well, I could add more because I think he's probably just forgetting more, and we've talked about this. He's not going to hospital every 4 weeks to get his exchanges. That's a big deal. He actually had his port taken out. I think it's going to be a year next month. So that's a big deal. And we haven't had you complain about pain. I mean, the only time I had him complain about pain when he had to exercise at school, and so he had done some exercises. It was more muscle related stuff, but he hasn't complained about like crises like pain.
I'm so glad. Do you have advice for, if somebody else was thinking about going through this or, had already kind of signed up to go through this? Do you have advice now in hindsight that you would give to somebody else?
Yeah, like find something to distract yourself, like for the whole time, and interact with like the nurses and stuff because they're nice, and yeah, that's pretty much all. Well, that's at least what I did.
That's really good advice. Something to distract is really important. Did you have something that you liked to distract you when you were in the hospital?
Games. That's what I used to distract me the whole time.
Like video games?
Yeah.
Yeah, that's what I remember from when I came by your room. And that's a common one. I totally agree. It's great to have something distracting. How about from the parent perspective?
Since he was 6 months old, we were in hospital every 4 weeks. Yeah. We were in hospital every 4 weeks, and that's all I can remember. I remember all the crises he had. I remember the one time that they thought he had a stroke and he was intubated at that time. He literally, the best way I can put it, stopped breathing. And I remember one of the nurses telling me to call my pastor and so that they can pray over him because they thought he wasn't going to make it. So, and then at the age of, I think around 6, he got the port, and then life changed for us. And he would resume life like any other kid. He would go to school. We were in hospital, but we were just there for appointments, not because of crisis. And then now we are here where he doesn't have to go to hospital. He's home. I mean, of course, COVID has changed stuff, but our life has changed a lot. I feel that there's more sort of confidence in just him having just a normal life as a teenager, being able to play, being able to even go out in the snow and not be like, no, I don't think you should go because maybe you'll get a crisis. I mean, we think about that, but it's not like top priority. It's not something that we are like so worried about. So I would just encourage the parents who are thinking to go through this. It is scary at the beginning because there's the fear of the unknown. But hopefully, as you see us sitting at this table, it's changed our lives a lot. There's the health part, and then there's the financial part too, for us as parents, because the frequent hospital visits, people may not know, but it does cost a lot of money. It does cost a lot of money depending on what insurance you have. So it's changed our lives. It's changed our lives for the good.
Our third interview features Dr. Stacy Croto, Medical Director of the Boston Hemophilia Center, who is the principal investigator leading the Hemophilia A clinical trial at Boston Children's. Dr. Croto will interview John Brissett. John received gene therapy in April of 2018. He was the first patient at Boston Children's Hospital to receive gene therapy for hemophilia. For John's gene therapy, is an in vivo gene therapy, which is a little bit different than the other two we talked about. And then with in vivo gene therapy, the gene and the vector are injected directly into the body through an intravenous infusion. So here are John and Stacy.
Thank you, Colleen, and thank you, Gene Therapy team. It is such a pleasure to be here with all of you today to celebrate Rare Disease Day, as well as the 10th anniversary of our gene therapy program. So John, your experience with gene therapy goes back a bit further than many. Tell us a little bit about your first introduction to gene therapy for hemophilia.
It does. And I just first want to say thank you for including me. I loved hearing the prior patient stories, and I'm happy to share my own as well. Yeah, so actually for me, it was the second experience with gene therapy. I was part of one of the initial hemophilia gene therapy trials back in the late 90s. And I went through that gene therapy trial as well. And it was proven safe, but not necessarily super effective. And it never progressed to a phase 2. And so there were a couple of other studies that were happening at that time as well in the late 90s, and they all had kind of similar results. And it was a different technology and one that was not quite as reliable. And the perception was that it was not quite as safe. And so I think after those studies kind of didn't progress, it was kind of a blow to the hemophilia community. And we kind of thought that, that was the end of it. We tried, and gene therapy probably wasn't going to be anywhere in the near future. And so when I started to hear about trials happening, in 2016, 2017, 2018, it really sparked my interest again. I was like, oh, this is happening again. There are new efforts in this area. And I was happy to see that the efforts were starting up again.
And so how did your first experience impact your interest for future gene therapies with respect to thinking about some of what elements you mentioned in terms of safety, effectiveness? Did that really increase your interest in participating in clinical trials and this is a modality, or was there some hesitation there as you thought about this next round of opportunities for gene therapy in hemophilia?
I think for me, the choice is always, and my hope is that whatever solution that is being investigated, the first thing is the safety, not necessarily efficacy. So if I can help to prove that these trials are safe, I'm happy to throw my hat in the ring. And I didn't have my hopes up with this study at BCH because of my prior experience. I knew that it was safe, but I wasn't expecting necessarily any sort of, or wasn't banking on any sort of clinical effect from it. If I could help to prove that it was safe, and just keep that process moving forward, that was my main goal. Of course, for me, I'm happy to say that it worked. And since that time, I've not had to have a single infusion, where for the first 40 years of my life, I was having three infusions a week for my entire life. So for me, it was definitely a game changer. But going into it, I didn't really have any hesitation there.
So for this most recent gene therapy, this was an AAV-based hemophilia gene therapy, so an in vivo gene therapy. Can you tell us a little bit about what the experience was like receiving this gene therapy? Was it what you expected, not what you expected? What was the process like for you?
Yeah, I mean, like one of the previous stories mentioned, I think the most the most intense or anxious part of it was just the initial antibody screenings. And I think if I remember correctly, the first round, I had like a trace amount of the antibody, didn't think that I was going to be able to participate. And some of the protocol changed, and I was allowed to participate a little while after that. So that first step where I was kind of screened out was really disappointing. And I was like, I really wanted to be able to participate in this. And then when that changed and I was able to actually participate, everything was like really smoothly. And then on actual dosing day, it was just another infusion. Like I said before, for me, I was somebody who was giving myself 3 shots a week for nearly 40 years of my life. And this was just one more shot. And it was really the last shot that I would have to have for quite a while. And yeah, I mean, it was just an hour of literally you, myself, and several other members of the team just watching this hour-long infusion and watching it drip with anticipation and excitement. And then once it was over, that was it. And then it was just a waiting game. So I remember that day, it was just a whole lot of waiting and watching and just wondering what was going to happen next.
Since we heard about from some of the other gene therapy participants, their gene therapy involved staying in the hospital and sort of that kind of close interaction with the team inpatient over the course of weeks. With our in vivo gene therapy, this was, as you highlighted, just an infusion, an outpatient infusion. And then at the end of the day, you went back home. But can you talk a little bit about what the days and weeks immediately after that infusion was like for you? We certainly didn't send you off and forget you. There still was a lot of interaction. So what was that like? And was that more burdensome than your infusions, or about what you expected?
It was about what I expected. I mean, it certainly tapers off after a while, but for those first few weeks after the infusion, there's a whole lot of blood tests that happen, and just making sure that the levels are being tracked really effectively, levels being sent off to multiple labs. So a lot of tubes of blood being collected. So for me, at most it was like a mild annoyance. Like, okay, I've got to go in, they've got to draw like 10 tubes of blood, and I'll see them again in 2 days, or I'll see them again next week. But there was nothing, it really wasn't any sort of annoyance for me personally, because it was part of the process. Like we were creating history, and we were all in great anticipation to see if those levels were the factor VIII levels were going to maintain, if they were going to drop off, how high were they going to get? Oh, this week they're higher. I'm at 30%. As a severe hemophiliac, my factor VIII levels, what prevented internal bleeding for me, were never above 0%. And to come back every week, have these check-ins where my levels were being taken and my levels were 10%, 15%, 25%, it was mind-blowing. Never expected that to happen. So for me, it was, there was some anticipation every week about, what's my level at? Have I gone up? Have I gone down? So it was kind of exciting.
And like you mentioned, you sort of approached this gene therapy opportunity experience with cautious optimism. What was your biggest hope and biggest anxiety in participating in gene therapy? And did it meet your expectations in the end?
Yeah, I think my biggest anxiety was something would happen where there might be some side effect or some safety issue that might stop the study. Again, the efficacy was great, but it was a secondary goal. So for me, just reinforcing the fact that the study and the technology being used was safe, and that other people, my story might help other people feel confident about enrolling in a gene therapy trial like this. That's my ultimate goal. So for me to be here today and say that, after an hour-long infusion, my life, completely changed, quality of life incredibly improved. And if anybody's thinking about doing something like this, like for me, there was no hesitation. And I'd say go for it.
So if you could offer advice for those that might not have the same experience that you've had in the past with thinking through gene therapy, the risks and the benefits, what advice to someone considering gene therapy would you offer?
Yep, I would say do your homework and weigh your pros and cons and think about your quality of life. I think especially for the hemophilia community and the sickle cell community, and I'm sure the ALD community as well, quality of life is really an issue. I don't want to see any kids that have severe hemophilia or sickle cell have to go through the things that I went through when I was a kid. And I can tell you standing here today that my quality of life completely changed, completely improved. And I would say do your homework, but enrolling in something like this is going to ensure that one day no child's going to have to deal with the issues that I had to deal with when I was a young hemophiliac, or somebody with sickle cell had to deal with when they were going through it. So, I'd say do it.
Excellent. Thank you, John, so much for your insights, for your candor. And we'll toss it back to you, Colleen.
Thank you, Stacy. We'd like to share a brief video with you now highlighting members of our gene therapy community.
Debido a la terapia génica, hoy puedo compartir el fútbol con mi hermano. Gracias. Because of gene therapy, I can play with my brothers. My name is Brian Rojas. I was diagnosed with adrenoleukodystrophy disease when I was 4. Because of gene therapy, I can ride my bike and play outside. Happy 10th anniversary. Gracias a la terapia génica, hoy puedo estar con mi familia y jugar a la pelota, que es lo que más me gusta.
Because of gene therapy, Paul could play at the park without a bubble or worrying about wiping something down.
Hey.
I'm Harry.
I'm Harry.
I'm 5 years old. I'm from Vietnam. Because of gene therapy, Harry is now active and healthy. Thank you.
Thank you.
Bye.
Bye.
Because of gene therapy, I haven't had to go to the hospital every 4 weeks for a blood exchange. And I feel like I have a lot of more energy, and I haven't had to go to the hospital any time around because I was sick or anything, and I feel like I'm doing a lot better. Thank you, Dr. Esrick and the team.
Debido a la terapia génica y al esfuerzo de mis médicos y colaboradores, logré continuar con mi vida, lo cual me hace muy feliz. Agradezco a Dios y a ustedes por su gran apoyo y feliz aniversario.
Nosotros como padres estamos muy felices por lo que hemos logrado. Feliz aniversario.
Hi, I'm Rachel and this is Kate, and because of gene therapy, Kate has an opportunity at a life with more mobility and independence.
Thank you. For many of us, that's a walk down memory lane, a happy walk, I may add. And now we have a few words from our founder, Dr. David Williams.
Thanks, Colleen. Thanks, everybody. It's really emotional for us to see this kind of video and to have all of you here today. I have to, first of all, call out Sandi Fenwick, who didn't mention this, but has been our leader for many years at Boston Children's, and is retiring soon. She's been a tremendous supporter of science over the years. I've had the privilege over the last 5 years to be the chief scientific officer at Children's, and in that role have met with her, I think, weekly or nearly weekly, Sandi. And I recently told her just last week, in fact, earlier this week, that I had the privilege over my 35 years in medicine to have worked for some outstanding chairs, physicians, but never have I had the chance to work with someone as good as she is, as effective as she is as an administrative leader of a complex institution and a supporter of science. And we're going to miss you a lot, Sandi. I also just want to give you a little background about our program and myself to put in perspective what you've just seen. So I actually started working in the field in 1982, if you can believe it, when David Nathan, who's my lifelong mentor, still at 92, sent me to MIT to learn about this newfangled approach called gene therapy, using viral vectors to insert genes into cells. And so my postdoc project was actually to develop a method to put genes into hematopoietic stem cells with the idea that someday it might be used to treat human diseases. And it was a very difficult postdoctoral period for me, but I've been privileged to be part of the field ever since that time. And subsequent to that, some of you may remember that there were severe complications in gene therapy for SCID, also called bubble boy disease. And those were complications in Paris and London, leading to leukemias in several children in those trials. And so a group of us around the world got together at that point and began to work together to try to improve the safety of those vectors while maintaining their efficacy. And that led to a consortium called the Transatlantic Gene Therapy Consortium. And when I came back to Boston from Cincinnati Children's about 15 years ago, I had the great privilege to meet Sung-Young Pai, who was already mentioned, and also Gigi Notarangelo, who was in the immunology program at Boston Children's. And the three of us had this vision that we could use the vector that we had developed as part of this consortium to start a trial in Boston and around the world. And in fact, that was the first trial in the bubble boy disease approved by the Food and Drug Administration after the leukemias that occurred in the London and Paris trials. That trial was very successful, and you saw in the video the first patient to receive that new vector in the world, and he's still doing quite well all these years later. And so all this has a tremendous personal meaning for myself, but I think what's really critical for everyone to understand is how much it is a team effort. And we could never have done it without not just the gene therapy team that's represented here and you've been introduced to, but the whole team at Boston Children's. The children also need care from many other subspecialties, and the infrastructure that we need to do that is quite complex, and we need really help from many of the specialists that aren't part of our team formally, but certainly help us. So that's really important for people to understand. The environment at Children's with respect to translation of our science is world-class and unparalleled in my view. And that's why it was a great privilege for me to come back 15 years ago and to help start this program. Now, I also, just before we go on, we've left a lot of time for questions, and there's a few in there that we want to answer. Once again, to thank everybody that's participated, and particularly the families and the patients. We understand how scary it is to enter into these first in human, first in children trials, and the progress of the field is dependent upon people like yourselves who are willing to do that. So we call you out especially for that. I also just want to make a note that this particular anniversary was really organized by Emily Williams and Dominic Malfara, who didn't get recognized earlier, but they put together the video and they put together this whole anniversary, and they've really done a great job of helping us communicate our science out across the many different populations or many different people that would be interested in it. So thank you, Emily and Dominic, for that. So I'm going to just now field the questions. I'm not going to answer them all myself. I'm going to field them out. So keep on your toes, panelists, because they're going to come your way, I think. I'll do some myself, but I'm going to pass them along. So the first question is regarding the latest Bluebird Bio news about adverse events in their sickle trial. Does this influence the ALD trial in any way? Is this the same vector used in ALD gene therapy? We knew that you would have questions about this event that just got advertised basically by a news conference about a week ago. So we anticipated the question, and we're in the process of sending out letters to everybody that's been in our trials to try to help educate everyone as to what this means. So for those of you who haven't heard, in a sickle cell trial run by Bluebird Bio, which is a foremost company, biotech company in the gene therapy field. There's been a patient recently who developed acute myeloid leukemia, type of leukemia. I can tell you we're in constant contact with the folks there, as well as the FDA and as well as the people at the National Institutes of Health. What we don't know yet, and Bluebird doesn't know yet, is the relationship of the vector to the leukemia. So that's still needs to be sorted out. So on the one hand, we can't a priori say that has no relationship to our trials that use lentivirus vectors. But on the other hand, I think it's until we know the relationship between the vector in their trial and the leukemia, it's a little hard to know whether there's a relationship or not. I would say in general, when you think about side effects of these vectors and in these trials, there's a component related to the vector or the vehicle in which the gene is being transferred in. There's a component that has to do with the disease itself. So every disease is a little bit different, and how the disease affects the side effect is a little bit different. So what happens in sickle cell trial, even though it's a similar vector, may not be relevant at all to the ALD trial. There's a component that's related to the way a patient is prepared for the gene therapy, the conditioning regimen. And I think we're not clear yet, and Bluebird is not clear yet as to how all three of those are potentially influencing the development of leukemia. Having said that, I would say the FDA has not asked us to put any of our trials on hold. We have put our own gene therapy for sickle cell trial on hold temporarily for enrollment, but that was not dictated by the FDA itself. So at this point, I would say there's not enough information that suggests that we should be particularly worried about the ALD trial or the other trials that we have going. Of course, whenever you change the DNA, as we do in gene therapy, there's risk involved in that, and we're sensitive to those risks and are following our patients, as you know, quite carefully for those things. So let me go to the next one. Once approved, will gene therapy be used as a preventative treatment for all boys diagnosed through newborn screening to eliminate Addison's disease and the risk of ALD? This is a great question. I think I'll ask Christy to field that one.
Absolutely. Thank you, David. I did notice through the chat there are a lot of questions about ALD, so I'll try to answer a couple of them as I answer this one. And I also noticed in the participant list that there are a fair number of our patients who participated on the trial and their families with us. And so just regarding the prior question, if any of you would like to speak with me or any number of our gene therapy program about what Dr. Williams was just talking about, please reach out. So addressing ALD and gene therapy, right now, there are no plans to move that into a preventative measure, but we don't know what will happen in the future. And I can tell by reading the chat, there are a lot of people who are really versed in ALD here. So just to refresh those to educate those who aren't, and that children who are born with ALD, those boys have a 40 to 55% chance of converting to the cerebral form of ALD. And that's what we use either allogeneic stem cell transplant for or autologous gene therapy. I noticed further down in the chat, someone was asking the difference between stem cell transplant and gene therapy. And so there are lots of differences, but probably the important one to highlight is in allogeneic stem cell transplant, you're using cells from another person that are given after conditioning chemotherapy. And in autologous gene therapy, we're using your own cells. Those are genetically modified and infused. Both of those treatments still use chemotherapy, and chemotherapy is associated with multiple different side effects, which is part of the reason we don't preemptively or preventively try to treat boys who have newborn screening positivity or those who haven't developed the cerebral form. we are still learning so much about boys who have been diagnosed through newborn screening, about the different types of presentations. And I think that the way we treat boys in the future will change, but right now we're not in a spot where we're going to treat anyone preventively just because of the risks associated with both therapies and not knowing who's going to progress to the cerebral disease. There are tools in development. Someone mentioned also in the chat the MRI perfusion studies. They're helping us to identify patients perhaps earlier. And I think that that's a really exciting step, that we may not have to wait till someone has a full-blown lesion or other changes, that we may even be able to identify boys earlier on. And so not yet for preventative therapy, but we'll watch the science as it evolves. And still as of yet, either allogeneic stem cell transplant nor gene therapy, neither of those treat the Addison's disease or the adrenal symptoms. And those are all, the goal is really to address the cerebral progression.
Christy, just while you have the forum, there was a question about the adult onset. Maybe you could.
Sorry, I missed that one as well.
Yeah.
Yes. So right now, there's, I guess, the question of adult onset cerebral ALD versus AMN. So I think the adrenal myeloneuropathy, the group knows that, or many in the group know that gene therapy or stem cell transplants not known to correct AMN, but there is an ongoing effort both in the US and Europe, a group that's focused on the adults with cerebral onset disease. Right now that there are no studies for gene therapy for adult onset cerebral disease, but this consortium of which we participate along with our colleagues at Massachusetts General Hospital are trying to understand the best way to do stem cell transplant for adults with cerebral onset disease, which may mean in the future, as we learn more about adult patients, that gene therapy perhaps could be an option in the future for cerebral ALD rather than AMN in the future.
Thank you. So there's a good question about, is it possible in the future for correction of other genes for gene therapy, other genetic diseases, I take it. So I would say that at Children's, with the help of Sandi and others, we've had a program in the last 2.5 years where we're trying to look at the genetics, the genetic causes of many rare diseases. And we have a program called the Cohorts Program, where we're looking at the molecular biology, the molecular causes of up to 30 different rare diseases. Our hope is that not only will improve our ability to diagnose these rare diseases more accurately, but in the end lead to new treatments of those diseases. And of course, one of the types of treatments we anticipate in those diseases would be gene therapy. So we're hoping it'll be a long road to that, but we're hoping that that will be the case in the future as we learn the genetic causes of many different rare diseases. Here's a question for Dr. Ezrick, which is what level of fetal hemoglobin range are you seeing in the patients treated in the gene therapy trial? And can you talk about the differences between your trial versus the Bluebird trial? How are they different? So Erica, you want to take that?
Sure, thank you. And thank you for the question. First about the question of fetal hemoglobin induction. Actually, I'm going to flip the order there for a second. The difference between our trial and the Bluebird trial. So we use a very similar lentiviral vector, but the actual target gene that goes in is different. So in the Bluebird bio trial, it's considered a gene addition trial in which a beta globin gene or a modified beta globin gene is added to the cell. And in our trial, we're adding what I call kind of a set of genetic instructions to target a gene that controls fetal hemoglobin. And the goal of that is to increase the level of fetal hemoglobin, which is a healthy, non-sickling kind of hemoglobin that all of us make as babies, and usually goes down to a very low level in older children and adults. But we would like it to remain higher because it's a healthy hemoglobin for people with sickle cell and prevents sickling. So that's the primary difference. There are some other specific differences in the makeup of the lentiviral vector that I won't get into the details about here. And in our trial, the majority of our patients have between about 20 and 40% fetal hemoglobin. There's some variability. If we did just recently publish our experience to this point with our first 6 patients, which I'm happy to share with you the reference for that later. and maybe Emily can help us with that. So our patients achieve approximately that range. And the fetal hemoglobin is pretty broadly distributed, which is also really important to look at. Again, happy to discuss more offline too.
Great, thanks, Erica. So this is a question that we can get a consensus on. Is it okay for children who have undergone gene therapy to receive the COVID vaccine? So I think in general, our approach to vaccines are that once you get through the immunosuppression part of your therapy, then you should get the vaccines that are appropriate for age. And that would also be the case with the COVID vaccine. So you have to have specific discussion with us, with your own child, depending upon how long it's been since they got immunosuppression, so busulfan in most of our trials. But those, in general, we would want children to get these vaccines as soon as they're eligible and can get them. But I would urge you to talk to the investigator in your particular trial so that knows your child to give you specific advice. So there's a question on, this is someone who says, I'm a father of a boy with DMD. He's 4 and still didn't start the steroid treatment. Is the gene therapy close enough so I don't need to start the therapy? So he has, I'm assuming DMD is Duchenne's muscular dystrophy. And I think we're still a ways from having that therapy being effective. So I would make sure that you go through with the current therapies that are appropriate for that with your doctor. But also ask your doctor about the eligibility of your child for any upcoming DMD gene therapy trials. This is a question, let's see, we've already answered the questions about the difference between gene therapy versus stem cell transplant. We can come back to that if people like in a minute. But the next question is, what challenges have come up over time as you follow those long-term, if any, with various trials? So the challenges long-term, I think, are not necessarily anything that wasn't anticipated to this point. So the questions with all the trials, because this is so innovative, so new, is number one, the safety long-term. That's the first thing. And number two, the long-term efficacy of the therapy. So both of these things are things that we're following, as you know, probably for long-term. So we have a long-term follow-up trial that goes out for 15 years after gene therapy. And so those are the two things that we continue to follow. I think challenges long-term are no different than those two things, basically. So then this is from a person in the UK. Sorry, my screen's jumping around a little bit. Let's see, this says, while known and unknowns and unknown unknowns persist for gene therapy, do you need to think differently about true informed consent in a trial setting and consent in a commercial setting to protect the interest of patients and their families, i.e. mandatory educational programs and validated knowledge assessments and risk-benefit tools, etc.? Yeah, I think we're, and I'll let anybody else who wants to jump in also talk about this, but from my standpoint, we're constantly thinking about informed consent. And in fact, many of you know that in our trials, we've consistently used what's called a consent monitor. So this is a person not connected with our trial who, after the consenting process, then meets with the family to make sure the family actually understood the consenting process, because these are quite complex trials. We recognize that. I think with respect to commercialized products, in general, that will require educational material because, of course, once an FDA-approved product is a product, then there's no consenting process per se. It's a matter of physician to patient education with respect to the side effects and potential efficacy of that particular product. I'll let anybody else on the panel who wants to add into that chime in. So feel free, any of you.
Yes, I would add in that engagement in medical decision making continues to be really important. And I think particularly in areas such as hemophilia, where in addition to gene therapy options, we do have a number of other successful, albeit a lot more challenging treatment options in terms of burden of treatment. And so while we don't have formal processes generally after a product is commercialized, I do think that, especially with respect to hemophilia and some of the other bleeding disorders who may shortly have gene therapy options coming down the pipeline, engaging in these conversations with providers that are experienced in gene therapy and hemophilia and bleeding disorders, and going through in a thoughtful way a non-clinical trial, but still informed consent discussion, and having time for reflection and assessment of patients and families' understanding of the process, the potential limitations, the both short-term and long-term risks continues to be really important.
I think that's true. I obviously fully agree with that. And I think one of the things that we think about as a program is that the informed consent in that process, whether it's after something's approved or on trial, really does need to reflect the disease, and that an informed consent for a patient with hemophilia may be different than an informed consent for a patient with ALD and sickle cell, and that we need to continue to engage with the patient communities, the patient advocacy groups. So it's a two-way street. So we're learning the best ways to approach families and patients. At the same time, you're learning about these therapies. So I think it's part of an ongoing conversation. And once we're after manufacturing, the consent process certainly is different, but we all really are committed to making sure that it is a full, robust, and educational discussion for everyone involved.
So,
I'd just like to add,
yeah,
oh, I'm sorry, Dave. I think I would just add that, as Christy just said, our informed consent process does not stop at the informed consent. And so that's an ongoing dialogue that we have with the patient every time we have a visit, anytime a new risk or a new benefit is introduced, whether it be in our trial or any other trials in the field. And I think, we're very transparent about that and wanting to make sure that our patients and families are very informed and have all of the most up-to-date knowledge. And I think that that's been our philosophy as well with in thinking about some of these products which are now and will be commercial in the future, is having a full educational materials available for patients and families so that they do have the most up-to-date information before they decide if this is the appropriate therapy for them.
I'm going to combine a couple questions and call on Christian Brendel, who's one of our experts in vector technology, which is sort of this, to paraphrase the question, is why are different vector systems used, some for in vivo delivery and some for ex vivo gene therapy? And maybe, Christian, you could take a moment to just explain those two differences.
Yeah, different vector systems simply have different properties which are related to the viruses they are derived from. And for ex vivo gene therapy, for example, it's absolutely essential to permanently integrate a gene into the genome to fix this, the component which was missing. This is not necessarily required, for example, in hemophilia, where a vector type is being used, which is very efficient in doing the job which is needed for this specific purpose. So we simply match the vector system to the specific disease, to fix the problem, the underlying problem.
Right. And the newer technology, which Dan Bauer, for instance, represents here on our panel, is to use not a viral vector at all, but to use different means to either insert to edit the genome itself. So rather than adding something, you actually change the sequence using very sophisticated new editing tools. Those methodologies actually don't use a viral vector at all, essentially. So that's the new sort of the new technology that we're developing, along with other companies are developing that as a new way of addressing. And it may be in the end safer or more efficient? We don't know until people participate in trials, just like the families on this call have done in the past. There's a question about Pompeii's. The question is there going to be gene therapy trials planned for Pompeii's? The answer is yes. Those are being planned, and we at Boston Children's will likely be a site for those trials in the future when they get going. There was a question about somewhere, I think, about when ALD will be approved. The plan is, I know because we're the lead site for that trial from discussions with Bluebird, that there's a anticipation that they will go to the FDA sometime in the current year or early next year to ask for registration approval for that. So that's a great news for us and for patients. And what diseases will these new technologies be used for? So I think that may be referring to the new gene editing approaches. Maybe Dan, you want to take a crack at that one?
Sure. There's great potential to apply the new tools to many diseases, not only rare diseases, but even to common diseases. Some of the initial applications will be diseases where there's genetic instructions that could be altered to have a beneficial impact, such as Dr. Ezrick mentioned, to increase fetal hemoglobin for sickle cell disease or beta thalassemia. But there's potential to fix the mutation itself at its core and reverse the disease altogether in cells. There's opportunities to add new genes to improve lots of conditions like immunotherapies for malignancies. So there's many opportunities to apply these.
So I think that's one of the exciting things for us in the field is that we see as the technology improves, that it's likely that the number of diseases that we can approach for curative therapy is going to expand, and we're all going to be very excited to be part of that. So I see we're at the top of the hour, and I want to thank everyone for your questions very much, and maybe turn things back to Colleen as our MC. Colleen?
Thank you, David. I want to thank everyone for joining us today. And if you have any other questions or would like to learn more about what we do, please visit us on our website at childrenshospital.org/gene therapy. Thank you, everyone. Thank all the panelists, to our team. I couldn't be prouder on a personal note to be part of such a great team. And to our patients and our families, thank you so much for all you do with us. Have a nice day, everybody.